Welcome back, everyone. So I will be the one moderating the half of the last session, I think, that we have for today, the first half of it. So I'll be introducing our next speaker, Dr. Belinda Nicolau. So Dr. Nicolau obtained a DDS degree from the Federal University of Paraná, Brazil, and a MSc and PhD degrees from the University College London. After working briefly with Paul Spite at UCL, she went to McGill to pursue a postdoctoral fellowship with Drs. Allison and Franco. She's currently a full professor and the Assistant Dean of Research at the McGill University, Faculty of Dental Medicine and Oral Health Sciences. And she's also an associate member of the Department of Epidemiology and the Department of Oncology, all at McGill University. She's the director of the McGill Summer Institute in Innovative Methodologies, and as a Canadian research chair in life course oral epidemiology, she leads a research program that investigates the etiology of chronic oral diseases using epidemiological methods within the life course approach. Please join me in welcoming Dr. Belinda Nicolau.
Thank you, Babatunde, for the nice presentation, and I'm sorry for my voice. So today I'm going to talk about a project that I recently started working. Was over the last 2 years that I started working with Fernanda and Roger, which addressed antibiotic resistance in dentistry, and I'm going to show you a comprehensive approach to evaluate knowledge, attitude, and antibiotic prescriptions in all Quebec dental schools, which we have 3 dental schools there. So I would like to acknowledge the co-leaders of this project, Dr. Roger Jones, who is actually the principal investigator, and Alice Lavigne, who is assistant professor in the Faculty of Dentistry at McGill. So as we saw some of the presentation this morning, MRR is on top of a global public health threat. It's among the 10 top global public health threats. And dentists, most of these prescriptions are from primary care, and dentists are responsible for up to 11% of the prescriptions. But these rates, they vary between different countries. And most of the data that we have thus far is mostly from high-income countries. So probably this number can change substantially. And we saw this morning, too, that there are a significant number of unnecessary prescriptions. And despite of the overall decline in antibiotic prescriptions, then dental prescriptions actually don't follow these trends. And you look at in Canada specifically, if you see here, there was between 96 and 2013, there was almost 13% decline on antibiotic prescription, but actually the number of dental prescription increased up to 62%. And so among the main factors that leads to this is the lack of knowledge. And a systematic review recently from 2019 showed that a high percentage of medical students display serious gaps in their knowledge of diagnosis and treatment of infection disease. And what is surprising is that some studies report 80 to 30% of the students believe that antibiotics are useful to treat viroses. And we see similar findings in among dental students. And also the literature suggests that this health professional inequities originate in their undergraduate years. Another factor that it's affected, that influence to prescribe in dentistry, is the propensity to prescribe when in doubt. So if the dentists are in doubt, they tend to prescribe. Another thing also is about the pressure to prescribe from patients, and this is very difficult because some patients, sometimes they want to have antibiotics. And other things that we must think as well is about insurance. So the dentists are nervous about being sued in case they don't, something happen and they don't prescribe. So then how can we address this problem? So within dentistry, the availability of data of students in clinical structures are limited, and most of these data are from surveys, which somehow is limited in scope. So we need to have approaches that are more broad in terms to try to understand how these prescriptions are performed. And so it's this that we try to do with this project. And so we set up, as I said, the project in Quebec, which is the French province of Canada. And in all 3 dental schools, we have 3 dental schools, 2 French and 1 English. And our idea is to examine the knowledge, attitudes, awareness regarding antibiotic prescriptions, and also to investigate the contribution of the dental curriculum and the role of different stakeholders in shape the vision of antibiotic use. To do that, we are using a sequential mixed method design. And I'm going to try to explain you what is this approach. This approach brings 2 very different philosophical assumptions in science. The quantitative approach is based on positivism, which is basically knowledge generated for observable, measure things, while the qualitative approach is based on constructivism, where knowledge is acquired from human experience and human interaction. So in terms of mixed method, we want to combine these 2 philosophical assumptions. As you probably can imagine, is a bit challenging. So just to give you another example of this, a concrete example of what is qualitative and quantitative. If you think about a quantitative research to describe a table, he probably going to say, oh, this is a square table with 4 feet. A qualitative research is going to say, this is an object where we sit, eat, and discuss. So in a very lay term, this is the difference between these 2 approaches. Let's see in terms of science how they are different. So in terms of qualitative approach, we usually use to understand the phenomenon to generate a hypothesis. In quantitative approach, which most of us are exposed to, we use to generalize, to summarize, to explain, to multiply evidence. In terms of questions, quantitative, usually we use open questions. We explore complexity, try to have a deep understanding of a phenomenon, while in quantitative, we just test hypotheses with specific variables. Data collection, qualitative, are small purpose, while in quantitative, we have representative large samples. In terms of data collection settings, in epidemiology or in experiment, we try to control everything to avoid bias, while in qualitative, this doesn't exist. And data collection, again, it's very rigid in quantitative, while in qualitative, it evolves as we go during the data collection. And is subjective and reflective, while the quantitative is objective. In terms of analysis, qualitative, we use words, images, and in quantitative, we use numbers. And so we, in terms of analysis, again, we just see the data and the categories that emerge from the data. in qualitative and in quantitative is statistical analysis. So in this project, now I'm going to talk to you about the first phase of it, which is part of it is already conducted. And this was a web survey, online survey that was designed by Roger, starting in Norway. And this questionnaire has 14 questions, which probes on perception and knowledge, experience and confidence, improvement and continuing education. The instrument was tested for reliability, was back translated to English, English to French, Portuguese, and Norwegian. And we also test the final constructor in terms of validity. And the study was conducting 5 places in 5 different countries. In Canada, in 2 universities. Here in Piracicaba, we conducted a survey. We also conducted in the south of Brazil and in Norway. So we saw from the results, one of the questions asked to the dental students, how important do you think the following challenges are in the world today? So as you can see, most of them mentioned that antibiotic resistance was among of the major challenges, and gender inequality also was common, except for Canada, that climate change was one of the most important. Another question is about if the students think that antibiotic resistance is an important topic for dentists. 88% respond that was. And surprisingly, although they said that was important, only 8.9% have heard about One Health. And here in these questions, they ask which area should be targeted to slow down the development of MRI. And again, they mentioned that the most important was antibiotic, the antibiotic in humans, and also public awareness. When we asked them the level of confidence, and these questions was asked only to the students in the last 2 years, the majority of them in different clinical situations, they said they were moderate to very confident to prescribe, except for one situation here that was slightly different. And here we saw about how do they feel about knowledge to prescribe in clinical practice. We see the majority say that around 7.5% average say that they have enough knowledge. And here we plot the high awareness. And those who have high awareness of antibiotic resistance seems to be more confident to prescribe in clinical practice. And finally, the last questions is the interest of the dental students in receiving further education and information on select topics. And you see that the majority of the students also want to have more information in different topics. So in summary, some of the reflections, we saw that our awareness towards antibiotic resistance, but there was lack of understanding about how human health and the environment are interlinked based on one of the questions. So there is some inconsistency here. Their high awareness is connected to confidence to clinical knowledge and supports the creation and maintenance of stewardship programs aimed to address MRI in dental schools, and dental students recognize the importance of MRI and need for continuing education. So this is the summary of the survey for the students. And now I'm going to talk about the second thing that we are going to do. We haven't started yet at this project. We are going to look at the dental records at the 3 dental schools in the past 5 years to see how the antibiotics are prescribed and try to understand, to see any patterns of this prescription. And this is, I mean, we just got funded for this project, so we are, at the moment, we start getting ethical approval to conduct this. And then the second phase, that is the qualitative phase, is this part now. What we are going to do, we are going to conduct with the GMG students that we run the survey, we are going to conduct focus groups, which is a technique for data collection and qualitative research. And basically, you have a group of students and you discuss, you have a structured interview where you discuss the different subjects that about you design an interview and you discuss the subject. And after that, what you do, you just create codes, and you, based on the teams, and you have 2 ways of analyze the data. One, you use a framework that has teams pre-established, or you can use also an inductive coding, which is just novel themes that come from the interviews. We also going to do in-depth interviews with clinical instructors, deans, associate deans, and people, academic administration responsible for the curriculum. And we are going to use the same kind of analysis, using thematic analysis, using a framework, but also using an inductive approach. And finally, what we are going to do, we are going to do a comprehensive analysis of the curriculum of the 3 faculties. So we are going to look at all the courses syllabus, how they are evaluated, and see how this is taught at the university. So I give you a brief overview of how we are going to collect all this data, but how we are going to make sense of this, because you can imagine that this is a lot of data, and this is the most challenging thing in mixed method. So it's this part here. So what I give you a few, these are really hypothetical examples just for you to understand how this methodology works. So let's say that we find in the quantitative analysis that the students who have high levels of knowledge about MRR have similarly high confidence to prescribe antibiotics appropriately. The qualitative show that students who consider themselves to be highly knowledgeable about MRR feel confident to prescribe antibiotics, but are unsure how to address overprescription when it occurs among colleagues. So this is an easy one to combine. The results of both components, they converge. They associate knowledge about AMR with the confidence to prescribe antibiotic appropriately. Another situation that we can have is, for example, very common, we can find the uniqueness on the qualitative when you learn from the interviews that you can capture additional dimension, or, for example, hesitancy by students who identify knowledge and confidence in their own prescribing practice to address overprescription by colleagues. And a last example, hypothetical, is when there is a divergency between the 2 components. Dental records indicate prophylactic use of antibiotic in dental school does not follow current guidelines. And in the qualitative, clinical instructors argue that guidelines are not clear and that prophylactic use of antibiotic must be determined on case-by-case basis. So then we need to find a divergent results. There is a lack of consensus on the best practices, and even whether it's possible to establish guidelines without taking into account specific patients' characteristics. So this is a bit one of ways to analyze. So what we do once we get the results of that. So the idea is that, knowledge and confidence, if we found that knowledge and confidence are highly connected, therefore this emphasizes the need for solid teaching and reinforcement of AMR throughout dental school curriculum. Another thing that we can also say that is specific stewardships training, how to address AMR with colleagues and patients. Open online courses like the MOOC. Clear guidelines, to develop clear guidelines to provide dental associations to distribute to the dentist, and campaigns to educate the population. So I hope I had given you an idea what this project is, and I would like to acknowledge my collaborators and the different teams in the different countries. Yeah, and thank you very much for your patience to listen to me.
Thank you very much for the presentation, Dr. Nicola. I wonder if we have any questions from the audience. Okay. Oh.
We don't have the ball. The cube is gone. So, since you're being streamed, maybe I should hold the microphone. Yeah. Thank you so much, Belinda. These are very important studies to conduct to find out how prescribers actually feel about their prescription practices. And you're actually finding a lot of interesting data. So congratulations on that, and thank you for a very good presentation. I was wondering, do you have any plans to look into why the prescription rates increased that much amongst dentists? 62% in a few years, that's a lot. Could it be that we are delivering different kind of services? Could it be that patients are more aggressive, expecting success and going to sue the dentist more often? Could it be that dentists are more overworked and have to just do something to get the patient out of the chair? I'm now speculating, but my question is rather, do you have any plans to follow up on this to find out why?
We are discussing this with, I talked to Pedro today about this. We are planning to look at this in Canada, but also here in Brazil, to look at how this prescription have changed over the years and trying to find reasons. I mean, in Canada, usually, because the system is a public system, it's not as, there is not as much pressure in terms of suing as there is in the US. So I'm not sure if this is an issue, but I know that the guidelines from the different dental associations are really outdated. And from conversations with different heads of clinics, I can see that. So I mean, we don't know, but we are on the planning to look into that. Thank you for the question.
Do we have any more questions? Going once, going twice? Yeah, that's it.
Thank you.
Okay, so I will be moving on to the next presenter, Dr. Achal Dariwa. So Achal is a PhD student at the University of Oslo. He is a bioinformatician with training in omics data analysis and software development. is primarily interested in understanding the effects of antibiotics on the microbiome and resistome development in preterm babies using metagenomics. He's very passionate about reducing the gap between data generation and data analysis by developing user-friendly and interactive tools to facilitate data understanding and hypothesis generation. Please help me welcome Dr. Achal Dariwa. You have the floor.
Thank you, Babatunde, for a great introduction. Thank you. It's in the, no, it's Matthias. I'm not sure the way. Thank you, Pedro. Good afternoon, everyone. Hopefully everyone had a great lunch. So today I'm going to talk about a tool that we developed as a part of my PhD, which is mainly aimed in doing the visual, statistical, and exploratory analysis of this AMR metagenomic data. So from the morning, we had some great presentation where almost all speakers, starting from Fernanda to Dr. Sharmila to George, John, they talk about metagenomics and how you analyze the data, how you characterize the diversity of resistant genes, the abundance of resistant genes in using the metagenomic approach. So I'm probably going to talk a bit more about metagenomics, like how actually you do this. The one thing that is common among the speakers that you guys have noticed or not, they are not just biologists. They are expert in the bioinformatics too. They know they have a very good training in bioinformatics, and so for them, things are way easier as compared to not everyone is a bioinformatician. You are on the other side, one side or the other side. There are very few people which are kind of in between, which knows both. So in this presentation, I'm gonna address two big challenges that is in the field. One, of course, the elephant in the room, AMR. The second is that you will realize what is the second one. So we know AMR is a global threat. It's not only affecting humans, it's livestock, environmental. So it's a complete chain of interaction, and it's not only us, but it's the whole environment that is getting affected. And according to the report, even though this is old, but they have estimated if the current trend of AMR supposed to be the same, we expect more than 10 million deaths, which will oversee the number of death which is caused by cancer by 2050. So what is resistome? So basically we all have microbe in our body, in environment, in animals, and the collection of all the resistant genes that is present in this microbiome is known as resistome. So as you can see here, it can be a various type: intrinsic resistant genes, proto-resistant genes, silent resistant genes. And how do you, so the first question that arises, how you characterize all the resistant genes that is present in the whole microbiome? So traditionally there was an approach called culture-based approach, where you culture this bacteria in the lab, and then you characterize these resistant genes. But sooner or later, people have realized that most of our microbiome you cannot culture in the lab. And that's why you need to have a technique which is not rely on culture dependent, and that led to the evolution of metagenomics, where you don't rely on the culture-based approach, but you just extract the DNA from the sample and you just sequence everything. So there are two approaches. One is sequencing-based approach. Another one is functional metagenomics approach, where you ligate this fragment of DNA into a vector and then phenotypically characterize these resistant genes followed by sequencing. So metagenomics now, it became a viable surveillance tool. And it has been used in multiple large-scale studies where they have studied for surveillance in not only in environment, but human, but also in livestock and animals. And what made this metagenomics a surveillance tool? Credit goes to next-generation sequencing. The sequencing costs have gone down immensely. And this has led to increase in the catalog of resistant genes or databases of resistant genes. So we can use these databases to characterize the genomes and metagenomes and find resistant genes in this in our microbiome. As you can see here, the sequencing cost, it goes exponentially down, and the number of resistant genes or class of resistant genes, it goes significantly up. Another game changer is development of bioinformatics or computational resources. There are so many softwares that have been present now which help you to characterize and identify resistant genes in genomes and metagenomes. So this is a significant development that happened over the past 10 years. So how do you do this metagenomic-based studies? So if I have to simply define, there are 3 steps: sampling, sequencing, and data analysis. So basically, let's say I have to study human resistome. So I collect the sample. It can be saliva sample if I have to study oral resistome. I collect this sample and go to the lab, extract the DNA from this sample, and send this extracted DNA to sequencing. And then the job of bioinformaticians start to analyze and make sense out of this data. So what actually bioinformaticians do? So once you send this sample to sequencing, what you got out of this data is raw sequencing reads, and this is a big data. It can be in GB and TB depending on how many samples you sequence. What you do of this reads is you basically pre-process this reads. For example, if the sample is coming from human, it is important to remove the human DNA from the sample if you have to characterize the oral microbiome or oral resistome. So you pre-process this raw reads to a high-quality reads using bioinformatics softwares, and ultimately you end up with high-quality reads. Once you get this high-quality reads, there are 2 approaches. Either you align these high-quality reads directly to the databases, resistome databases, which contain resistant genes, or you assemble these reads into longer quantities and then followed by annotation with the resistant gene databases. So in both the approach, what you ended up is having an abundance matrix, as you can see here. So what you can see here, you have this gene identified in the sample, and its abundance across each sample. So it's basically abundance. Once you get this matrix, the next objective is to make sense out of this matrix. Because sometimes in this matrix, you can have 1,000 resistant genes across 100 of samples. So it's not easy to make sense out of this or to understand it. And the analysis in which you make sense out of this count tables is kind of known as the downstream analysis. So, well, easier said than done. There are some challenges when you do this kind of downstream analysis. If I have to categorize the challenges, it can be statistical challenges or databases or visualization related challenge, which I'm gonna talk a bit. So the first challenge, I don't think so I will go more into it, but I think Dr. Baker already covered why this sequencing data is not absolute, it's a relative data. So basically what you expect, and this is what you get out of the sequencing data. So you need to use proper methods or proper normalization technique if you have to analyze this data. The second or very common problem with this kind of data is, depending on how deep you go sequence the sample, you either characterize more resistant genes or more microbes, and if it's like less sequence, there's a chance that you might miss something. As you can see here, if it's a deep sequence, you basically identify 5 genes. But if you do the shallow sequencing, you miss one of them. The third challenge with this kind of data is there's a lot of zeros in the matrix. By a lot of zeros in the matrix, I mean you cannot use normal parametric or non-parametric tests with this kind of data because the distribution is normal, not normal, or not even like it's a kind of a binomial like it's a very different distribution, negative binomial distribution. So it's not, you cannot use the normal test that you use for other kind of data directly on this kind of data sets. We know there are, I think Dr. George already tell about, like, there are various databases where you have this sequencing for resistant genes. But the issue with this field is these databases are inconsistent. The gene naming in this databases, the annotation information about the resistant genes, the class, the mechanism for resistant genes, they vary from database to database. So you actually cannot compare the resistome profile of a sample characterized through one database and then through another database. Another problem that I want to highlight, unlike the microbiome data, where one species can have one genus can belong to only one phylum, or one family, or one family can belong to one phylum. Here the challenge is different. So the annotation structure is acyclic, unlike the microbiome data. Another big challenge is these databases have direct or indirect information about which ARG is present in which organism. So it's a kind of a multiple association or multiple relation. For example, one gene can be present in multiple species, and one species can carry multiple resistant genes. But does these databases provide this information? The answer is no. You cannot say Klebsiella pneumonia contain this number of resistant genes, because they don't provide this information in the databases directly. Now comes to the overall challenges with the analysis. This data is complex and big. Not only complex and big, you need to have bioinformatics skills, which all of our presenters had it, but it's not the same for everyone. And the third and most important challenge is we are not standardized. There's no one best method that works for all. So it's kind of exploratory that you have to explore of multiple methods to analyze your data. And after talking about AMR resistome, the second challenge that the biology is facing, the overexplosion or overburden of the data. I know Anirban told me that it's good for bioinformatician to have more and more data so that we will get our jobs. But yeah, it's true. But at the same time, as of now, we are living in a big data era. The main issue is not to generate the data. The main challenge is how to analyze this data. And that's what we are lacking. The most important, another gap, there's a growing gap between the data generation skill and the data analysis skill. And this is growing from the college itself, because this is how we are educated. We are a molecular biologist, or we are either a bioinformatician or an IT guy who's like taught separately. But the thing is, I think it is important to actually fill this gap if we have to improve or if we have to advance very quickly in the field. And this applies to every field in biology, I think. So in order to fill this gap, the one way is to develop this user-friendly interactive tool and giving power back to the wet lab people or physician or clinician to analyze their data itself without knowing any computational skill or bioinformatics skills. And that was the main reason why we developed a software called Resist Explorer. So it's a web-based software. You don't need to have programming skills to do any analysis, but I think you can do a lot. So if I have to divide the workflow of Resist Explorer, it's basically 3 steps. You input your data, which can be a list of resistant genes, or it can be abundance matrix characterized from the metagenomic sample, or since we know metagenome not only characterize the microbiome profile, but also the resistome profile. So you kind of integrate this and answer some questions like, whether bacterial phylogeny shapes the resistome or not through integrative analysis. And then once you input your data, you have data pre-processing where you annotate, filter, or normalize the data. And then using visual analytics, you can generate several graphs or try to understand this data through exploration, intuitive exploration. So this is one of the features. So as I told you, the issue is like these databases have direct or indirect information about the species carrying this resistant genes. So we thought, why not to present this information in terms of a network, which is easier to understand. So as you can see, the circular node are the resistant genes, and the square nodes are the species which these resistant genes are found. The interesting thing is, like, as you can see, most of these species that carry these resistant genes belong to phylum Proteobacteria, which makes sense because they are known to be notorious bacteria or having these resistant genes a lot. So basically, with this kind of network, you can have a lot of information, or you can test various hypotheses about your list of resistant genes. The second thing we had in the software, we provide comprehensive visualization support for resistome data. So I think this is the common figure that you find in every resistome publication or every microbiome publication, that you use composition stacked bar plots to show how the microbiome profile or the resistome profile of the sample is. But along with this, we have provided various other visualization method also, such as Sankey diagram, sunburst diagram, Sankey diagram, and this is tree map. And these are all intuitive, so you actually can click and go out, go in, zoom in, according to the level of analysis or according to the level of annotation level that you want to analyze in your data. Just like microbiome data, where you not only analyze at the species level, but you go to the genus level or phylum level or class level. What we had also support for other type of analysis that is common in ecology or microbiome analysis, where you do the alpha diversity, beta diversity analysis, clustering analysis. And as Dr. Baker already said, in this software, we don't only provide the traditional approaches, but we also provide these methods which are basically designed for compositional data, called CODA, the compositional data analysis methods. We also provide comprehensive statistical analysis support for resistome data. So if you have to identify which are the genes which is differentially abundant between two conditions, which can be a healthy patient versus a disease patient, so we actually provide 5 methods, which is DESEC, HR, LDEX2, and COM. Some of these methods have been used in the other omics study too, but some of the methods, for example, NCOM and the LDEX2, they are specifically designed for the compositional data. The logic behind providing multiple methods is there's no one method that works the best. There's a paper from, I think, from Canadian University where they said it's not one method that you can choose. You have to try multiple methods to gain the confidence, which is the differentially abundant gene in your sample. So that's the reason why we provide multiple methods. What you can do also is you can do machine learning algorithm. For example, random forest and support vector machines to classify your samples on the groups based on the, like it basically computers unsupervised clustering or unsupervised classification, as you can say. What we have provided also support is like you can find association between the microbiome data and the resistome data. So you can answer questions like whether microbiome shapes the resistome, or there are some bacteria which co-occurs in the same proportion as some resistant genes. So you can kind of do the correlation network across the microbiome and the resistome data generated from the same samples. So just to conclude, this is Resist Explorer is a one-stop shop and valuable One Health AMR metagenomic-based bioinformatics resource. So it enables research and clinician with no bioinformatics background to actually analyze their data in an interactive and intuitive manner in real time and using a variety of standard as well as more advanced method. And we believe this kind of development of tool reduce the gap between data generation and data analysis skills. So right now, this tool have been used by 2,500 users across 64 countries. And it's been already published. And we have also a massive online opening course where you can actually read more in detail about this, how this software works, if you want to try. So I would like to acknowledge my all co-supervisors who are all there, Professor Fernanda, George, and Ulf, along with my whole Dr. Peterson lab and the funding agency who supported this work, along with the organizer who provided me this opportunity to be in symposium and present my work. And not only to them, I would like to thank especially Dr. Pedro. You are the Superman, and we are inspired from you. So, along with Fernanda and Nancy to organize everything and make our stay and everything so smooth. And of course, the Resist Force group, you guys are awesome. It's been always, like, I don't feel like I don't know any of you from before. And last but not the least, the student in Unicamp. You guys are the best. I haven't seen a bunch of people who are so friendly. Thank you for all the patience that you guys shows to us, even in selecting a pizza, whether it's a vegetarian or non-vegetarian, but you guys are awesome. So, obrigado. And if you have any question, I will love to take it. Thank you.
Thank you very much, Acha, for the lovely presentation. Are there any questions? Okay, Rita. Okay.
My question was just, you mentioned pre-processed data.
Yeah.
So what's the data input that you actually need to use this platform?
So basically, when I say pre-processed data, it's basically the count abundance metrics. This is where we start the downstream analysis. But the aim in the future is to actually basically start from the raw sequencing data, so that it would be. But the thing is, like, there are so many already available bioinformatics pipeline that help you going from the sequencing data to generating the abundance metrics. What we are lacking in the field is going from this abundance table to allow you try different methods on your abundance table without relying on a bioinformatician or, yeah. You have to anyways rely, but at least you can explore a lot on your own.
Okay. So if there are no more questions, I have a question myself.
Sure.
If the moderator can ask a question. Okay. So this is a very fantastic work, and I love the fact that you're democratizing this kind of skills.
Yeah.
But I do have a bit of a question about the other side of this. My experience is that once you have many of these tools that simplifies, everything, then what usually happens is that, folks without maybe necessarily the experience would subject their data to these kind of analysis, and they might start, pontificating on what those means without reaching out to the necessary expertise. So at what point does a regular user put a phone call to someone like George, that, oh yeah, I'm out, I don't have these skills, I can't interpret this? And do they have that same, if they continue using the tool, would they have some sort of a false sense of security that they know what they are doing? And is that being addressed, or are there...
I mean, that is yeah, valid point. That's one of the question that we got from the reviewer. Like, why are you providing multiple methods of statistical analysis? Whether user will use the best method wherever they will find the statistically difference, and they will choose this method as the best method or show that method. But again, that's not the logic behind reasoning. Of course, if you have to manipulate the data, you can manipulate from the day one. Like, you don't even have to sequence. I can generate the count table the way I want. I can show beta-lactam is going up in a patient without antibiotics, but that's not the point. Like, yeah, I agree. That's ultimately you have to go to the bioinformatician, because for sure they had an experience. They know which methods works the best, and you cannot, like, you can for sure use the information or the results that you got it here as a starting point, but probably you have to call George to get the right information, whether you're interpreting right. Yeah.
George should not be afraid of going out of work anytime soon.
Yeah. Yeah. Thank you. Thank you. So basically, we have this Google forum that we are using right now to user Google Groups.
User base increases, people can keep on posting their queries, and already experienced users can give the
Can answer. Yeah. So yeah.
we choose between this.
Yeah. Yeah, that's the thing, because
Community is the best answer for this time.
exactly. So basically, this is what happened in MOOC. There's one user. She published 2 papers on the same tool, which I haven't even done that, but she kind of used the tool and she published already 2. So when we launched the MOOC, she was the one who was answering the other users, oh, you have to do this in this tool. So I was like, that's good. That makes sense. Yeah, yeah.
Thank you very much.
Thank you so much. Thank you.
Hello, everyone. My name is Ian McIntyre, and I have the pleasure of introducing the next 2 speakers, or the last 2 speakers of today. First, we have a presentation from Dr. Pedro Ricomini. He's a professor here at the University of Campinas and will be presenting biofilm control and antibiotic resistance.
It's here. Sorry. It's here. Aqui. Now I think that I'm ready. Okay, so we can move on. So first, thank you, Ian, for the introduction. It was nice, so I think that you were worried before, so now it's done. That's great. I would like to talk to you a little bit more about some strategy of biofilm control that goes on the topic about antimicrobial resistance, and maybe is a possibility how to control biofilms without using antimicrobial substance. So I'll just start my talk
talking about biofilms, it's the area that I work more, so it's an area that I really like about it. And biofilm community is like biofilm, when you think about biofilm accumulation, so it's a community of microorganisms that's characterized by the cells that bacterial cells or fungal cells that they are adhered to a surface and they are massed in polymeric matrix of polymeric substances. So when we think about biofilm, we are not talking only about a lot of bacterial cells, in the past was thought that they were like accumulated as a plaque. We have like a live structure that is growing, so they are. We don't have only bacterial cells, but we also have the polymeric substances that they are like entrapped along the cells. So here is like a figure of biofilm. So we have the cells and we have the matrix among the cells. And the matrix, it's important to keep the biofilm organization and to protect the cells as well. So in this matrix, so among the cells, we have DNA, we have polysaccharides, so they are polymeric substances that they are present or molecules that they are present, and it's important to keep this biofilm structure. And when you think about how to kill bacteria, how to kill a fungi, we are all thinking about to use some substance that kill the cells. So maybe now we can change maybe the way to control the biofilm, not only targeting the bacterial cells, maybe it's not possible to kill all of them, but it's possible also to reduce this biofilm accumulation or accumulated already. So when we think about the human body, we have like very different sites. So when we think about biofilm accumulation, of course, we have some sites that the bacteria, they can accumulate easier, but others not. So when you think about the body, we have like different body sites. We have different environments, like different temperature, different pH, different oxygen concentration, and also different nutrients. So all this information, they are important because they are going to make all these characteristics make the site proper for some specific bacteria to grow. So we have like in our body different sites with different bacteria accumulated on different sites. So we don't have the same microbiota in our whole body. And when we think about the microbial colonization, so we have some different molecules and structures that favor this biofilm colonization as well. And we are going also to have like different constituents of the matrix, because if you think about the nutrients that we have and all these characteristics that they are specific for each site, so we are going to have different matrix constituents that can be like produced by the cells, or can be also be part of when cells are dead and they are like the inside components, they are going to be outside as well. So a question that sometimes people, they are used to say that, biofilm we have to control like that or like this, and so is a way to think about like if biofilm control is a strategy or if it's a unique strategy for all types of biofilms. So like we have like different sites, different characteristics, so we are going to have different biofilms. So the answer is no. The biofilm control strategy is not unique for all types of biofilms, so we have to think about it. And when we think about health and disease, considering the human body, when we have bacteria, it's not like it doesn't not mean that that we have some unhealthy condition. So the health, when we say about health, it does not mean that we have to kill all the bacteria even like when we consider the oral cavity, like it's not possible to kill. Maybe we try like to do some mouthwash, use some mouthwash to kill a lot of bacteria, but just after we are going to have bacterial growth, colonization, so it's not possible to kill all of them. So they are still growing, they will keep growing. So it's not possible. So kill the absence of bacteria in this case does not mean that you have a health or unhealth situation. Like when you have oral diseases, the oral diseases, they are mainly caused by the biofilm accumulation. When you say caries, periodontal disease, of course, is not the unique thing that's necessary to have, but you need biofilm present there. When you think about it, it's not possible to control the disease just by killing all the bacteria. As I mentioned, the bacteria, they are there, they are going to grow and grow again. So the microorganisms will have to live with them in balance. So they can be our friends as well, and they are good to our body as well. And like the biofilm, as I'm telling you, we are going also to have it inside our mouth. So when you consider like the hard structures that we have in the mouth, so when our tooth start to erupt in our cavity, so you have like a solid surface, and different from the epithelium, from mucosa, is not a shedding surface. You have a hard surface, and this surface is easily and rapidly covered by some proteins that form a pellicle that covers that structure. And then the bacteria starts to adhere and to grow. So the biofilm is a very live condition. And this is an example of some bacteria that they have adhered to a surface, and they start to grow, and the biofilm starts to accumulate. Here's an example of a biofilm of just like 24 hours. And when you think about how diverse is the bacteria that accumulated, it's not like just the same way when you think like, oh, if I get a sample of biofilm, everything will be similar. The amount of bacteria that they are sparsed on that sample is the same. So it's not. They have like some groups of bacteria that they are important one each other, so they start like to grow. So here it's possible to see some images of fluorescence in situ hybridization, and it's possible to see that different colors means like different groups of bacteria, and they are growing. So it's not possible to see that is a regular or a singular way of organization. And that is important about the biofilm when it accumulates and it grows. And when you think about biofilm, one thing that is very known about the dentists, but of course people from other areas that work with antimicrobials, is that when you have like the biofilm formed, so when you have antimicrobial exposure, like it's not easy to kill all the bacteria that are inside that biofilm, because you have like a structure that favor that only the outside bacteria may be killed in some minutes, but these structures also favor that the bacteria that they are inner of this structure, they can survive. So even like if you can kill some bacteria outside, the inside bacteria, they are still alive. So after a while, the biofilm starts to grow in the same way. So as I'm telling to us, it's not possible like to kill all the bacteria in the mouth to have a health condition. And it's interesting when we see that, because when we see like a lot of studies that a lot of antimicrobial substance has activity considering like planktonic bacteria, oh, it's like 100%, it kills all the bacteria. And when we use the same substance trying to control biofilm, try to see how it behaves, how it acts, oh, maybe the results is not so interesting. And or it's not possible to have like a good results that's possible to see or to observe an effect that the substance has killing the whole biofilm. It's very difficult to find this situation. So, like an example of that, it's a study that was conducted, sorry. It was conducted by the ACTA group in Netherlands, and it's possible to observe here, like you have biofilm accumulated. So in green, you have the green cells, so the cells, they are alive. And the B picture here is possible to see how the bacteria, they are there after being exposed to chlorhexidine. So it's possible to see that the upper level of this biofilm structure in red, that the bacteria started to die. But when we look like inside this structure, like all the bacteria, they are still alive. So it's very difficult to control the bacteria or the microorganisms in the mouse or the biofilms just using like this strategy. And when we think about dental caries is more specific also when we consider the metrics, because dental caries is always related about the frequent consumption of sugars. So mainly sucrose here, when like we have sucrose during this biofilm formation, so we have like a more thick, a bulk biofilm that's rich in extracellular polysaccharides. And these extracellular polysaccharides, they are dispersed, they are entrapped in among the cells of the biofilm. And like this extracellular polysaccharides, they are not degraded. So the biofilm will be stable until this biofilm is not disorganized, is not mechanically or like we are going to see enzymatically disorganized. So here, like it's possible to see like a amount of biofilm. It was a children that we follow in a discipline, a clinical discipline, and it's not possible even to it's possible just like looking to the over the teeth that you have like a huge amount of plaque accumulated on that surface. So we tried to stain, but it was not necessary. But we have like a lot of plaque of biofilm accumulated, and when we see this thick biofilm like this, like very bulk biofilm, it's possible to see that this biofilm probably has a lot of extracellular polysaccharides in its structure. And how just as a clue, how these extracellular polysaccharides, they are produced and what are the main and what could be a target when we try to control the biofilm. So we have some bacteria, mainly S mutants like group. They have like glucosyltransferases, and they can synthesize glucans. So just example here is like in yellow the enzyme, and here you have like the sucrose molecule. You have the glucose, the glucosidic bond with the fructose. So the enzyme, this glucosidic bond is broken, and now this glucose molecule is transferred to another glucan that is being like constructed, synthesized. So at the end, you are going to have like a huge chain like of the glucose molecules that they are putting together each other, just growing as a big chain. And the problem for us is that like this one, when you have the alpha-1,3 glucans, because we don't have enzymes in the mouth or some bacteria that has some enzyme that is able to break this type of glucosidic linkage, so to degrade this type of glucans. So they like still be accumulated. So a strategy could be to break this type of glucans. And when we were talking about this structure, so it's totally different when you have when you compare only when you compare like sucrose, how the biofilm is grown. You have sucrose as a substance, as a carbohydrate that the bacteria can use to ferment, of course, but to produce the extracellular matrix as well, rich in extracellular polysaccharides. So here it's possible to see that the characteristic of this biofilm is totally different when you form the same biofilm when you have only glucose and sucrose alone as monosaccharides. So here it's possible to see that there is no extracellular polysaccharides formed, and here you have a lot of extracellular polysaccharides formed. And here, like the microscopy image, is possible to see as well, and here, non-formed in green. On the cells and green, and the red part, that's extracellular polysaccharides. So it's totally different when you have sucrose, when you consider the biofilm formation. And here's just some more examples that is very, it's possible to see very easily how these extracellular polysaccharides, they accumulate among the cells. And of course, it's different and it's important when we talk about caries disease. And like here, when we grow a biofilm in this in a similar condition, just like here when you have glucose and fructose, and here when sucrose is present. It seems like, looking at the first picture, it seems that, of course, you have some cells in green. And here, if you look at the picture just below here, it seems that you don't have a lot of extracellular polysaccharides among the cells. But when we remove the green channel, so that it's possible to see the amount of extracellular polysaccharides that are present in this biofilm structure. And it's interesting because the biomass, or the amount of the volume that this structure builds or forms is totally different when you consider the glucose biofilm, glucose and fructose biofilm, and sucrose. So you have a more thicker biofilm formed in that condition. So we are talking a little bit about this, how the glucans, they are present and they are synthesized by this enzyme, because this enzyme has a specific activity for this substrate, and only sucrose can be used as a substrate for this extracellular polysaccharide synthesis. So we have some interesting characteristics of this biofilm. We have the stronger adherence to the tooth, so the bacteria easily accumulate on the tooth, and the carbohydrates can penetrate and be fermented in this biofilm. These acids can remain for longer periods near to the tooth surface, leading more demineralization. And these polysaccharides may also can have as a barrier against antimicrobials, if you think that some antimicrobials could be used to try to control some part of the bacteria that are present in this biofilm. So one strategy could be to break. I'm just talking to you about some strategy that could be used in order to control we are talking about dental biofilm, but it's possible also to use in other biofilms as well. So you have the glucan chain. So if you have enzymes, specific enzymes that we don't have in the mouth, and the extracellular enzymes, it's possible to break this glucosidic bond, so we start to destroy or to disorganize this biofilm. And it's possible also to use DNases. It's known that the use of DNases has also some activity disrupting early-stage biofilms. And we did a pilot study. I'm going to show the results to you. So maybe DNase and alpha-1,3 glucanases, the enzyme that's possible to break, that is present in other species of bacteria and fungi, not present in the mouth. So maybe they combining these two bacteria, maybe we could have or optimize the enzymatic activity degrading this biofilm matrix. So here we are talking about a different strategy. So instead of trying to kill bacteria in order to control the disease, we are talking about to try to degrade the matrix in order to control the biofilm. So here's our target, the matrix. So this pilot study, we just use it, we conduct other studies, but just as a clue for you to understand and how the results we got. We use like glucanase and DNase in a biofilm formed with Streptococcus mutans, that is a bacteria that has the glucosyl-transferase, so is able to produce a lot of glucans, so we have the substrate to be degraded by the enzyme. So it was like a study that we formed biofilm for 24 hours, and we added 1% of sucrose, and we had 4 treatments. We have like the control, DNase, the glucanase that degrades alpha-1,3 glucosidic bonds, and the mixture of these 2 enzymes, DNase and the glucanase. And we evaluated the biomass using a crystal violet method that we stain this biofilm and recover, and we also evaluated the amount of extracellular polysaccharides that was present in that biofilm. So in a very short way, the biofilms were grown, so the medium was discarded. We washed it, we did the enzymatic treatments, and then we stained the wells with the biofilms with crystal violet, and the remaining dye was removed in order to measure, like if the enzyme removes more biofilm, we expected to have lower amounts of crystal violet staining remaining inside the wells. And after that, we just got we treated the wells with sodium hydroxide to remove the EPS, and then we just precipitated this polysaccharide and quantified. And we got interesting results, like DNase, like when you see this graphic, is the graphic related with biomass. So this is the percentage of reduction of absorbance that we got. So lowered, so higher the results, it seems that more biofilm was removed. And here, like we have like the best combination, like the combination of DNase and the glucanase, we had like higher results. And when we compare like the amount of the reduction by the DNase and by the glucanase, so it's possible to see that if you compare the isolate results of each enzyme, and when we have the 2 enzymes together, it was like 35% higher than the isolate results. So it was possible to see that a synergetic effect when the both enzymes they were together. And so it was possible to see also when we evaluated the extracellular polysaccharides concentration or reduction actually in this graph, it's possible to see that of course DNase, maybe some of the extracellular polysaccharides could be like removed, but not by the enzymatic activity, maybe related more with some activities near like the cell wall. I think that's the thing that we have to study as well. But when we have the enzyme glucanase, we got a lot of reduction, and both together a higher result as well. And it's interesting here that when we have the glucanase, it's possible to see that the reduction of EPS was very high. And when you think about the biomass, it was not so high. And here, when you have the link of the combination of these 2 enzymes, the concentration was the reduction here of biomass was much higher. So maybe we still have some, maybe like having the 2 bacteria together, the 2 enzymes together, it was able to disorganize even the matrix, like the extracellular polysaccharides, but may also have some link about the DNA that's extracellular, and some a link with the cells and bacterial cells that could remain, like we saw here, that we had lower results. So this data, like we observed that when we had this combination, so it was like good for the when you think about strategy to remove or to degrade this type of biofilm. Of course, we have a lot of new challenges, so it's necessary to check the temperature and pH, how is the enzyme activity in those situations. We did that result, so they are working in our oral environment, so it's not a problem. How to deliver the enzymes, if we could use a gel or something like that. The salivary clearance, because the saliva is always being produced. Some students, they are there, they have listened that a lot. So it's possible to reduce or a mouthwash that you use. So if you have some enzyme there, probably it's possible to be removed with this salivary clearance as well. Oh, maybe how to keep the enzyme in the biofilm. Oh, sorry, too fast. Maybe how to keep the enzymes like linked or like attached in the biofilm, like keep working even after you like have done with the treatment. And of course, the oral cavity, like here we conducted like an in vitro study, so it's necessary to go further for like studies in the oral cavity and biofilm formed in situ or in vivo as well. So I would like to end my talk thanking the São Paulo Research Foundation for the grant, for the INTERPART program that we are working together with the University of Oslo as well. We have Mateus that is there working with Professor Fernanda. So it's very good to have this collaboration. Our faculty, our dental school, I would like also to thank Professor Igor and Annelise, the University of São Paulo. They are helping us with these enzymes, how to have them and how to obtain them, and as I said, the University of Oslo. So just a reminder that Uf has talked at the beginning that today we start the antimicrobial resistance week, trying to prevent like the resistance. So we are going to see a lot of campaigns during this week, and this symposium is of course part of a strategy that make like people think more about some other source or some other ways to control biofilm or the diseases, reducing the amount of antibiotics or antimicrobials used. Thank you very much.
All right, thank you very much for your presentation. It was
Okay, you are welcome.
very nice. And if anyone has any questions, please come down and ask them.
Oh. You have to use the microphone.
So you talked about this strategy that we can degrade the matrix, but we also have bacteria that are being selected in carcinogenic biofilms. And if they are dispersed in the oral cavity, they can reestablish a biofilm. So would you think that a combination of this treatment with antimicrobials would be effective?
I think that, of course, if you are able to reduce the amount of biofilm, like degrading the amount of biofilm that you have, and if you need to use some antimicrobial, of course, you are going to have like higher effect, or you are going to be able to kill like a lot amount of bacteria because you have removed some amount of biofilm. So maybe is a strategy, not, we are talking about like oral biofilm or dental biofilm, but this strategy can be like used in other sites of the body.
Do we have any other questions?
So I'll go down.
Oh, okay. Well, thank you again for your presentation.
I'm breaking the protocol. Sorry, okay. Thank you, Jan. Check here, read this presentation.
And for our final presentation today, we'll have Dr. Britta Rude presenting from the University of Chicago, Illinois. She is a postdoc in Dr. Federley's lab, and she will be presenting streptococcal quorum sensing systems, friends or foes.
Thanks for waiting for me. I'm the last talk of the day, so hopefully I can make this a little bit shorter so we can all leave. So, as Ian said, I am a postdoc currently in Michael Federley's lab at the University of Illinois, Chicago, and today I'm just going to tell you a little bit about our lab and the type of research we're doing on streptococcal quorum sensing systems. Hopefully. Okay. So why do we even care about quorum sensing in the first place? Well, quorum sensing is one of the ways that bacteria coordinate group behaviors, which is obviously of particular importance to many different facets of physiology with bacteria. So I just want to introduce you to this concept in general, since I'm not sure how many people really think about this on a regular basis if they're not a microbiologist. And so in general, what this types of systems use is a pheromone, and this pheromone will actually accumulate according with the growth of the bacteria in the population. This triggers some sort of change in gene expression, which allows for change in behaviors and phenotypes. So these quorum sensing signals are known to induce group behavioral changes, and they are really dependent on the bacterial species. Now, in particular, streptococcal quorum sensing systems rely on what we call RRNPP systems. So these systems, again, rely on a pheromone gene, which is triggered by some sort of sense of an environmental change. This pheromone gene is then produced, processed, exported into the extracellular space, and is imported by neighboring cells. This then binds a transcriptional regulator, which I'll explain about a little bit more in a minute, that we call RRNPP type regulators. This transcriptional regulator can then bind upstream of the genes which it modulates. So what are these types of regulators, and why do we call them RRNPP systems? So they're structurally characterized by two domains and a number of canonical members of this family. So this includes the RGG-COM-R, RAP, MPRR, PURGE-X, PLCR, obviously standing for RRNPP type regulators. And these are transcriptional regulators which are characterized by two types of domains. One is a helix-turn helix binding domain, which allows them to actually interact with the DNA that they actually regulate. And then, of course, the TPR-like domains, which actually allow for them to interact with the peptide signal. So this peptide signal is thought to trigger a conformational change, and just shown here is the structure of one of these regulators, COM-R from S. thermophilus, and then allow for the DNA to either be released or for it to interact, depending on the type of regulator you're looking at. So streptococci in particular contain a lot of these, and they regulate a lot of different facets of their physiology. And what I want to emphasize is that a lot of different streptococci that are frequently found in the oral cavity contain these regulators, and we haven't really characterized what all of them are doing. So for instance, the Federley lab has particularly focused on the RGG systems present in Streptococcus pyogenes. They tend to control facets of virulence. However, there's RGGs present obviously in Streptococcus mutans, which a lot of people study from a dental standpoint, as well as not shown Streptococcus ferus, which I'm particularly interested in, and I'll tell you why later. And of course, there's also been work done on a number of other RGG-type genes that are found in other species of relevance to oral health, or just health, human health in general. Now, streptococci often use these to regulate some not-so-friendly aspects of physiology from our standpoint, although maybe it's considered advantageous to the bacteria. So for instance, in Streptococcus pyogenes and in some other species of strep, this actually modulates biofilm formation. For instance, when you have the system off, you don't have biofilm formation. When the system is on, you get good biofilm formation. It's also been shown to be involved in invasive infections. For instance, another RGG type found in Streptococcus pyogenes. When you have this system present, you get these invasive necrotizing fasciitis type infections. However, when this system is absent, you do not have this invasive infection. And of particular importance, especially for the development of antimicrobial resistance, is the fact that COM-R, one type of RGG regulator that's found in a lot of different streptococcal species, actually is able to control competence. And competence is the ability to take up DNA from the environment and bring it in and incorporate it into your genome. And from an antimicrobial sort of resistance standpoint, this is of concern because streptococci are frequently human pathogens, and they're increasing in their antimicrobial resistance, as we've heard from many other people today. I'll just show you a few snippets. So for instance, Streptococcus pneumoniae, it's responsible for over 2 million infections in the US alone per year, and 30% of those infections are completely antibiotic resistant. And if you just look at data from the HWO, so this is from 2020, you can see that the percentage of penicillin-resistant non-wild type invasive isolates range anywhere from about 5% to 50% of the isolates obtained from patients. This isn't just Streptococcus pneumoniae, though. There's a lot of other species of strep that are relevant to human health that are increasing in their antibiotic resistance. This is just shown from a report from the CDC in 2019. And Streptococcus pyogenes is obviously the causative agent of strep throat, necrotizing fasciitis, scarlet fever, a number of different really nasty diseases. And you can see that over years from 2010 to 2017, resistance to erythromycin has nearly tripled. So they're using these to regulate a lot of different things that we don't necessarily look at as friendly to us, and obviously not so great for antimicrobial resistance. But they also use these to regulate biosynthetic gene operons that we might actually be able to use. What do I mean by this? Well, so in conjunction with our collaborators, we found that these RGG systems actually are frequently encoded upstream of what is known as a RAS-RIP operon. Now these operons, RIP stands for ribosomally synthesized and post-translationally modified peptides, whereas RAS stands for the type of enzyme that actually modifies the peptides that are produced from this locus. So in this case, what you have is this RGG transcriptional regulator that's induced by a quorum sensing type system. You then produce this precursor peptide. It's then modified by this RAS enzyme, which relies on a radical generated by S-adenosylmethionine. You can then transport this out of the cell, and it has various effects. Now, actually, streptococci produce a lot of these, and what we found is that, so if you look at, so we did our collaborators did a bioinformatic analysis of just all the different SHIP RGG systems paired with these predicted BGCs, RAS, RIPs in general. And what you can see here is that across different streptococci, you can see a lot of these species contain different sort of families that they've classified based on the conserved precursor peptide motif, and that these species can basically make a lot of different types of these compounds. And our collaborators and us have gone on to basically characterize these compounds structurally and actually show that these species actually produce these compounds as well. And one compound that I want to focus on in particular is this family called WGK, because this really is the only family that we've really actually characterized extensively as far as its biological relevance goes. All of these other compounds we have only really characterized from a structural standpoint, so we have no idea what they're doing in streptococcal physiology. Now, what does WGK do? Well, so we've shown that Streptococcus mutans and Streptococcus ferris can produce these compounds, which when we were deciding on the name, our collaborators wanted to name it something fancy, so they wanted to call it triglycins for the modification that's actually produced. Now, what we found is that these triglycins can actually inhibit the growth of other streptococcal species and actually modulate the growth of their producer species as well. These are produced, again, by one of these RGG SHIP systems that are actually regulate the loci directly downstream of them, producing this inducing this biosynthetic gene operon. And this functions as any other of these quorum sensing systems do. Now, in data, so I don't have time to show you all of the data that we published, but we've basically shown that these can inhibit the growth of very common oral commensals, such as Streptococcus mitis. And I'll just show you here some MIC type data. So you can see here that around 25 to 100 nanomolar of this compound that we call triglycin A can actually inhibit the growth of Streptococcus mitis, Streptococcus oralis. And we didn't stop there. We tested many other streptococcal species, and we saw that basically all of these species could be inhibited in some way, aside from, interestingly, Streptococcus pyogenes, and we don't know why that's not inhibited by this compound or a modified peptide. Now, of course, we were wondering, well, what sort of relevance does this have to the producer species? And so we asked if these compounds could actually modulate the growth of their producer species as well. Oh, so sorry. This is the opposing triglycin from Streptococcus mutans and Streptococcus ferris. So interestingly, although these compounds only differ by a cysteine to a serine, you can see that these compounds can actually inhibit the growth of the other species that produces them, even though they only differ by one amino acid. So that's great, but what about biofilm formation? Because just because you inhibit something planktonically doesn't mean you modulate biofilm formation, and this is obviously really essential for these species' life cycles. So we decided to just ask if it could just inhibit biofilm formation in general. So we took triglycin and added it to a very simple in vitro system. We'd love to do this in a system that's a little bit more biologically relevant, but just as a first pass, we decided to see, okay, does this actually have any relevance whatsoever? So we put it in a glucose condition, obviously sort of EPS minus versus a sucrose-rich condition, and asked if Streptococcus mutans could basically, if this compound could inhibit biofilm formation, and we saw that it did. Interestingly, however, when we delete this RGG regulator, we still see biofilm formation in these presents. And initially, of course, you might ask, well, is this just dead cells or something just lying here? No, they're actually forming biofilms. However, the architecture is completely different than the biofilms that they're forming in the wild type condition. And we're doing some more experiments to investigate this. And if we complement this back, so this is just providing PDRA in trans, you can see that we can restore this phenotype back to wild type. Now, of course, the next question is, okay, so it can inhibit the formation of biofilms. Can it disperse biofilms? And unfortunately, the answer is no. So basically, when you add it at a biofilm that's already been formed for 24 or 48 hours, you can see that this compound does not trigger the release of biofilm. So it seems to just inhibit biofilm formation at the start. So, of course, our next question was, well, what about Streptococcus ferris? Does this also respond in a similar way? Because it's the other organism that produces this. And people don't really think about studying Streptococcus ferris because it's a rather understudied organism. It's not typically associated with oral health, but it does produce rather high levels of this compound. So we were interested in studying it from that standpoint. So we found that triglycin, so Streptococcus ferris behaves very similar to Streptococcus mutans in that it forms thicker, more robust biofilms in the presence of sucrose, as it has a very similar physiology to mutans, although it is different in a few key ways. So what you can see here very clearly is that similarly in glucose and sucrose-containing conditions, the addition of triglycin inhibits the ability of these to form biofilms. Interestingly, however, though, when we delete the regulator of this system, for Streptococcus ferris, we actually see the inability of these species to form biofilms in glucose, and it doesn't really seem to affect the ability of this compound to modulate it in sucrose. So we don't really have a good explanation for that, and we're currently looking into it. Another thing that I want to notice, or wanted to bring up, is that we've noticed that Streptococcus ferris actually forms intense aggregates in the presence of triglycin A. For instance, if you look at it in a glucose-based condition, so here, one of the downsides of using this is, of course, the cells tend to settle to the bottom. And so you really do have to wash these biofilms out to get an accurate readout. But one thing that you can appreciate here is that as you increase the amount of triglycin, depending on whether it's with wild type or this delta PDRA stream, you see these increasing aggregates. And you see the same in the sucrose condition. So if you have no triglycin, you get this nice thick biofilm. But as you add increasing amounts of triglycin, you can see these aggregates start to form. So clearly these compounds affect streptococcal physiology, but what about their context in the oral microbiome? Could any of them be used as therapeutics? We need to really do some more investigation on what these could possibly do in the context of the host. So we are currently developing a study in conjunction with Dr. Chen and Dr. Fernando Peterson, who have agreed to be collaborators on this study. And we want to ask the questions, basically, how do these compounds modulate the oral microbiome? What's the mechanism of action of these compounds? And then what about these other compounds that we've identified? So we currently have a study that's basically based a lot on Dr. Baker and Dr. Edlin's initial studies published in PNAS. And what they did was they actually took saliva from subjects and pooled it, and they did a time course at which they looked at basically different conditions over time. So we're doing a modified version of this because it's a rather acute response that these species have, so we don't want to basically drag it out for 28 days. So we propose basically to just take either treat the consortiums with PBS or triglycin, passage it over a short period of time, and then examine how the oral microbiome changes in response to this peptide. So with that, I'd like to thank, obviously, the RESISPART group for inviting me to give this talk, as well as our collaborator at Princeton and funding from UIC and NIH. Thank you.
Thank you, Dr. Rood, for such a nice presentation. Does anyone have any questions for her?
Congratulations, Britta, for your talk. My voice is terrible. And I would like to know, it is easy to produce this compound or to obtain this compound? How, like...
Yeah, so that was one of course, the things that so when we initially started studying this, we had to produce it kind of heterologously in E. coli, but we've actually just been able to develop a protocol to purify it in high yields from Streptococcus ferris, so we can get milligram levels consistently. And so we're hoping to write that up and publish it. Yeah, so we can actually get a lot of this compound. No, I don't know. Maybe? Yes, I guess.
Sorry if I missed this, but any speculation on the mechanism of how it inhibits the biofilm formation in S mutans?
That's a great question. So I think it has something to do, I wonder if this compound is actually signaling in some way. So sometimes these RAS-RIP molecules can have like a secondary function in doing that. And so I kind of wonder if it's actually inducing some sort of gene modulation and changing how the cells are actually responding to their environment. But we don't really have a good idea. That's just pure speculation. Honestly, what a good experiment would be is expose the Streptococcus mutans to this compound and see how its transcriptome changes, just in a very short sort of way.
Thank you to both Pedro and Britta. I was just wondering, it's maybe the wrong country to make this question in. How about just removing sucrose from our diet? Will that help?
Of course. Yeah, of course. Obviously, eating less sugar will help in many different ways besides just the formation of caries and biofilm. Obviously, obesity, a lot of different things. I think one of the interesting things about this is just the ability of these compounds to prevent biofilm formation from starting. Obviously, it doesn't help if you already have biofilms formed, right? But preventing it from starting in the first place might be a good place to start, in combination with therapies maybe that can remove it.
Any other questions?
Oh. That's fine. Yeah. You don't have to only ask me.
Yeah. Okay. I would like to congratulate not just you, Britta, but all the speakers today. It was a great day. I would like to ask Fernanda and Ulf about what has been done recently about antimicrobial resistance that is working, what is really is done that works to help this condition, and what is not done that would be effective and in your opinion. So that's a question about the topic. But congratulations to all of you. The presentations were great.
Ms. Peterson, would you like to start to answer this question? Thank you for that question, and also thank you to the organizing committee. I think this has been a very good symposium, and I have enjoyed it tremendously. What has been done that has worked on the AMR? The first thing that runs to my mind is all these countries around the world that has put AMR on their political agenda. They have developed national action plans, and they have really put a lot of efforts into spreading the message that we need to work together to solve this. This is not an issue about polarization. This is not a yes or no answer. This is a question where we need to work internationally. We have to act locally, but we need to collaborate internationally to solve this. AMR is the environmental crisis in a healthcare perspective. And I think this is the most promising thought and philosophy that has been shared since Dame Sally Davies put it up there on the European agenda and spread it to the world in the following years. So I think the national action plans are important. I think also the reason that national action plans work better in some settings than in others are the same that we experienced during the pandemic of the COVID-19, the SARS-CoV-2 virus, that some of different political situations, different cultures, different nations have different ways of being loyal or trusting their healthcare or their governmental authorities. So I think it has a lot to do with trust, the way we adhere to any national guideline, any national advice, any national action plan. So I think these things are important. And another thing that has also worked kind of in the wake of all these national action plans and all this international collaboration is that we are, I think now, much more colleagues internationally than competitors. I think we are joining forces, sharing experiences, and developing better and better actions so that we can together jointly fight AMR internationally. Miss, would you like to supplement?
I would like you to say a few words to conclude our symposium. So first of all, thank you all for the active participation in our symposium. We are living in an area of major global threats, and a major one is antimicrobial resistance. So this is a field where internationalization is key and can benefit enormously from multidisciplinary approaches. So the talks today varied from public health, policy, high throughput technologies, AMR mechanisms, possible diagnostics, and educational gaps. So I hope this will bring lots of ideas and possibilities for future collaboration in research and education. And a lot is already happening. Pedro, you are really a superman, as Ashal said. And the Brazilian students and the staff are great and have really spoiled us again. So thanks all the brainstormers for your participation, speakers. And I would like to invite all to stand up to give a big applause to Pedro and all the others that received us here.